Hey guys, welcome back to another video by Biology with Xuan. Today we are going to do another complete guide video, which is the IGCSE Biology 0610 Alternative to Practical ATP Paper 6 Complete Guide. So this will be a slightly lengthy video. So what I want to do is that I have put timestamps in the description below so you can navigate. So but if you're a new person, don't know what is alternative to practical, this is a video that you should watch it entirely. But for those who are familiar with it and want to see specific parts where I give tips and tricks, the timestamps is where you need to do. Okay, let's start off the video. Alright, so what is paper 6? So paper 6, it's basically that's why it's called alternative to practical, meaning like you are not going to do a real experiment in exam conditions, but the experiment is being printed on a paper where you are just doing inferring and answering to questions that is being set. So it's a 1 hour paper. No additional materials are really needed, just a ruler, a calculator, an eraser, pencil, and a pen. It's a very skill-based paper, so this sometimes requires a little practice and knowledge, and it's 40 marks in total, 20% of our qualifications, no weightage to a final mark, and it's taken by both core and extended students, and it's all testing AO3 objective, which is experimental and all of these related stuff. Okay, so technically that's it. So the topics that are tested in paper 6 are quite vast. Sometimes it's considered repetitive. It's come up repeated in the past few years. So you must know how to measure volume gases of liquid, masses, temperature, times, lengths, and topics like diffusion, osmosis, food tests, rates of enzymes and catalyzed reactions, pH and use of the hydrogen carbonate indicator, litmus and universal indicator, photosynthesis, transpiration, heart rate and breathing rate, respiration, tropic responses, and then continued would be observation and dissection of seeds and flowers, germination, continuous and discontinuous variation between the histogram and the bar chart, use of methods of sampling, observe, record and measure images, make clear line of drawing a biological specimen, calculating magnification, adding labels, use simple apparatus in situations where the methods may not be familiar to the candidate. So all this is quite application, not just by memorizing, but you're also giving a chance to apply your skills in different kinds of experiment situations. Okay, so the specificity of the question tested. So we have known that there are different kind of topics that may be asked in here, but here are just some specific things that they can ask, like identifying, draw, complete a label diagrams, explain common techniques, most appropriate apparatus or method of task, and describing food tests, the pH of a solution, hazards and safety precautions, explain techniques to ensure the accuracy of observation and data, planning experiments and investigations, identifying the IV and the DV, describe how X and Y variables should be kept constant, appropriate number of range of values for the independent variable, justification, safety precautions, how to record results, process of results, and make reasoned predictions. And making record, observation, measurements, estimates, know how to take readings from apparatus, analog or digital, like from thermometers, balance, and take sufficient observation of measurements, qualitative observation for food and other tests, and then suitable tables, record observation and measurement systematically, interpret and evaluate experimental observation data, how to process data from maybe calculation, and then you present your data in a graph, and then analyze and interpret data, know what is interpolation and extrapolation, and determine a gradient or the intercept, knowing to justify from the graph itself what does it mean, and form conclusions and evaluate the quality of the observation of data and anomalies. Then lastly is just evaluate methods and suggest possible improvements. So they give you different kinds of methods and techniques. You must find the control, sources of errors, suggest possible improvements to the apparatus, experimental arrangements, methods, and techniques. Okay. So a general mathematical accuracy in IGCSE biology is that you need to, when you're recording a particular apparatus and its volume especially, you need to round off your nearest number to 0.1 centimeter cube. Okay, and then for thermometer, it's usually to the nearest 0.5 degrees, like 35.6. You don't give 35.612 or 35.62. And then time to the nearest second. No millisecond inside your answers. Okay? And question one. So question one, we would usually start off with the very long given experiment question, and of course it's happening in every paper 6. So what you should be doing here is that you shouldn't be skipping this entire instruction. You should be reading it closely. So questions that can ask using this is that they ask you to draw a table, which usually is about 2 to 3 marks. Recommended for you to get a full mark is by 2 columns and 2 rows minimum, and usually they may ask you to fill in data from the results they have collected, and record readings from ruler and thermometer or measuring cylinder, usually about 1 mark or 2 marks. Write a simple conclusion from the table you have drawn, like as temperature increases the rate of diffusion is faster. Identify one source of error, safety hazard, safety precaution, like hot water, and what happens if you never deal hot water properly, you will burn your hands. And improvement to reduce error, like thermometer, you didn't check the zero point before you start measuring, you need to check for 0 error. This for the improvement. Repeat 2 or more times so that you can find out anomalies. If it's numerical data, you are able to find the average. And IV, DV, CV happens across here. Okay, and graph questions. Graph questions nowadays in the 2023 exams is that they give 5 marks or 4 marks question. Why? They ask you to create a legend. Legend is like key signs, is that they give you a bunch of data. You must know how to classify them in 2 different kind of things so that you're able to put and draw your graph by separating from different data to not make it so confusing. So graph questions is certain in every IGCSE exam, and they may ask you to draw a line graph or a best fit line graph or curve, but usually a curve graph is quite rare in biology but frequently in chemistry. So points on the graph should be clearly marked as crosses or encircle dots of appropriate size. Okay, do not give any other kind of dots to represent your plots. And a graph should be drawn with a sharp pencil, including your labelings and everything. Make sure it's in pencil, do not write in pen. Each axis should be labeled with an observation with the unit appropriate: seconds for time, gram for mass, and thermometer for degrees, and percentage, the percentage symbol. And unless instructed otherwise, we always remember that the independent variable is always on the x-axis, and then the dependent variable is on the y-axis. I always remember the IVX, DVY. It's like this, for example, on the picture next beside it. Okay, and food test. Food test is also another one of the most certain questions that come out in exam, and you must be familiar because it comes out in paper 2, paper 4, and paper 6. So usually for food tests, you just need to know that iodine is testing from starch, where it's usually from brown to blue-black, and then Benedict, which is the reducing sugar test, where it comes from light blue to brick red. And then ethanol is usually from colorless to cloudy, and burette for protein is from blue to purple. But for one exception is that for reducing sugar test with Benedict, you must mention that you need to heat up with a Bunsen burner before you see the result. Okay? Question 10. Not question 10. I mean planning investigations or planning experiments. This is a 6 marks question that comes out every series actually, and it comes out in paper 5 and paper 6. So this is one example question from the beside, like fruit juice contains vitamin C. Plan an investigation to determine the effect of temperature on vitamin C concentration in fruit juice. You can already see that there is a clue that has been given, an IV and a DV has been shown. So IV will be the effect of temperature, and the DV is the change of concentration. So what the question needs is that they want to find how the IV is being handled, how the DV is being measured. 3 control variables needed to keep constant. Control experiment like setting up waters or anything. Repeat the experiment 2 or more times for a better reliable result, and also include a safety measure. And tips for planning investigation is I always use the mnemonic, I don't care so run away. IV, DV, CV, safety, repeat, average. Average only used when it's in numerical data. So IV, you know that it's something that you're going to change. What is it changing? So that for the question just now, we are going to change the temperature. You can see at least 2 different temperatures stated. Any temperature is fine, 30 degrees, 60 degrees, you get a mark. And dV state the dependent variable, state the unit, state how are you going to measure it. So for the concentration just now, we have the volume of number of drops dCPIP added, the constant volume until the color changes, and many more. And the CV is to state 3 different control variables, describe how are you going to control them. So example, since this is temperature, right? Temperature can change due to its environment. So what you're going to do to change it is by using a thermostatically water bath. So this is called an equilibration, to make sure that everything goes to the same point as you mentioned in your steps before you conduct the experiment. This is to make sure it's a fair test. And safety is to state a safety measure. What kind of safety measure? Maybe you could say that you may be using a sharp knife to cut something, and then you must make sure that you wear gloves all the time to prevent cuts. Okay, and then repeat at least 3 times. See, 2 or more, replicate, repeat investigation 2 or more times. And an average is usually when they give you numerical data and you must find an average after you have collected your repeated experiments. Okay. Alright, in paper 6, right, there are some common independent variables that can be asked. So like just now we mentioned about temperature, we use a thermostatically water bath. And the possible range can be 10, 20, 30, 40, 50, 60 degrees, whatever you want. And humidity is depending on, it's a transpiration question, and usually it's with or without a plastic bag, because if you put a plastic bag on, the humidity can change. And light intensity is usually by putting a lamp at different distances away, measuring using a ruler, maybe like 10 centimeter from the sample, 20 centimeter, 30 centimeter, that will be the IV. And airflow is usually like how much you want to measure the gas exchange and how the stomata is producing photosynthesis. For example, usually it's in the photometer, where you use a fan or don't use a fan. If you put fan, there's more airflow, probably more transpiration, because the more wind, the increase in transpiration. And carbon dioxide concentration, you use the different concentration of a hydrogen carbonate solution. I always remember the colors is from using PM ROI, purple being the lower CO2 concentration and yellow being the higher CO2 concentration. Alright, next one, percentage calculation. So every time in paper 4, paper 6, they will come out a percentage calculation. It's either percentage increase or percentage decrease or percentage change. So the formula for percentage increase is final minus initial over initial times 100%. If you don't know what I'm talking about, final minus initial is that when you see that 2 different values, right, the first one will be initial, the second one will be final. You have to read the question properly. And if it's percentage decrease or percentage change, it will be final minus initial over final times 100%. Then you, of course, you need to put the percentage lah, of course. But for percentage specifically, right, if you see like the decreasing, the first column is 132.4 and the second column is 127.4, and they ask you to find the percentage change, you solve, you should be getting 3.779 in your calculator. But however, you can see that the first column to the second column, right, there is a decrease in change, decrease in the numbers, for example. So when it is decreasing, you have to put a negative symbol before the value itself. So the answer would be negative 3.77%. Okay, if the number is increasing, you just don't need to put a sign. It's already known as positive number. Okay. Magnification. Magnification is coming out in paper 2, paper 4, and paper 6. It's always there. So you must know the formula is magnification equals the image size over actual image. And usually in question nowadays, they have one that is in millimeters and one is in micrometers. And before you start any calculation, you must make sure that you convert everything to the same unit, because if you never convert, you get be confused in which are you trying to use. Technically, always use in micrometer is always the best. And remember that magnification, when you find a number, there should be a times X sign before the value. And remember, you need to know that 1 millimeter equals to 1000 micrometers. Okay, next question, biological drawings. So there is always a drawing in paper 6. So this drawing, they always give you a specimen ready. They ask you to just replicate that drawing. Very simple, is that just draw a large shape, just copy its outside line and then just make it look similar but in a larger shape. So don't draw any details, just draw the outlines. Follow what the instruction is saying. Take care of any labels that are asked in the question. Remember, when you are labeling, do not use arrowheads. Use a ruler to draw a line pointing to the part and label it on the edge of the line. No shading. This is not an art class. Just literally just draw the specimen. Draw the drawing at least 50% larger, because they are giving you a very large space in your paper. You should be using a pencil and draw it perfectly. And don't use a ruler or compass to draw. Use freehand. Just make sure it looks natural. And read the question carefully to see any specific segment is required. Don't draw individual cells because it's going to take a long time, and don't draw the surrounding box of the image. Just follow whatever the instruction is given. Don't do over because you will get deducted. And lastly, that's all for this entire guide. I know I want to do more, but this is coming from my notes that I have made. I will link it down to the description below, and please check it out, read it before, and practice as much of paper 6 questions as you do, because you find that there can be a replication in the same type of question but it's been reworded to a different question. So check it out in the description below, and this is a free guide that I'm giving you all, and this note is pretty much also taken from that note itself, so you get a great idea in what am I talking about. So if there's any question, please write down in the description below, and make sure if you use the timestamps, just guide around to which one you want to see. Alright, so see you guys in the next video. Bye bye.